Hello. So now today we'll enter into this vectors used in the transformation of animal cell lines. See, animals are cells are nothing but means they are not like the plants or microbial cells. See, because if you take bacterial cells or prokaryotic system, the system is very simple, and there will be no much kind of resistance offered by the cells compared to the higher eukaryotic system. And even the organization of the genome all are very simple in bacteria. Whereas in case of eukaryotic system, if you take yeast or even if you take the animals or even the plants, the organization is going to become a bit more complicated at these particular organisms. And as genomic organization becomes complicated, at the same time the cells are going to show much more resistance towards the foreign entity. It can be even a DNA or a nucleic acid. When it enters into these particular cells, they have a tendency to break it down due to the restriction as well as the DNases, which are nucleases which are going to be there within the cell. So therefore, whatever the kind of transformation we want to get into these organisms, it need to be done with a specialized type of vector systems. And particularly if you compare between the yeast cell, plant cell and animal cells are comparatively easy to transfer. Why? Because the cell is not possessing any kind of cell wall. We have direct protoplast with us, so therefore easily we can obtain the transformation. But only thing is the survivability of that particular transformant is a key. And another thing is, like, in case we want to develop a particular stable transformant, so then we need to have a specific type of vector systems which have the capacity of integrating itself into the genome of the host cell. And due to the complexity of the host cell genome, sorry, so it is also going to be important in the process of transformation. The care should be taken, whatever the transformation means gene or the recombinant DNA which is being vector system we are using, so it should also have a capacity to be either even in the if it is going to be present as an episome, it should be very stably present. If it is going to be integrating into the genome, it should specifically integrate at a particular part of the genome or into a particular chromosome. So then only it will be very useful. Okay. See here, the commonly used vectors, like for example, there are people have used the plasmid vectors and lambda vectors, cosmids, pack, back, yak, all those things. So here these particular vectors, they have their own limitations with respect to the foreign DNA carrying capacity. But whenever we think of an animal cell, and as well as when we think of about eukaryotic gene, the size of these particular genes are going to be compared quite high, and therefore whatever the vector we are going to use or choose, it should have the high DNA capacity. And also we have to concentrate on the copy numbers and their ability to integrate it within the genome of the host cell. So here if you see the plasmids, they are multi-copy plasmids, up to 20 kilobase they can carry. Lambda vectors, example bacteriophage lambda, you can say they can carry up to 30 kbase. Cosmids, they can carry up to 40 kbase. Packs, they can carry up to 80 to 90 kbs. Bags, 100 to 300 kbs. Whereas yak, which is having 100 to 1000 kbs capacity, and means a very large DNA carrying capacity is there for the yak. So in the transformation are in developing the cell lines with the animal cells, stable transformants or even transient, or people have actually used different varieties of vectors. And by understanding, see, when we want to go for transformation of large DNA molecules like 100 to 200 kbs, then we can go for bags or yaks. But when we go for bags or yak, we have a problem, that is these particular artificial chromosomes, they have a means they are not so much stable, and there is a chance that they can get disintegrated into the host cell when they are going to be subjected for, means when they are being used for transformation. They are very, very susceptible because of their larger size. They are not going to be much more stable in, means physically stable. So there are, see, these kind of things are going to pose a problem in using them as a vector system. And also, in order to do the transformation, specialized methods are going to be required for doing the transformation because of their larger size. So that's why people thought of using simple vector systems to achieve this particular goal and look for other vectors. So during which people also try to think of altered plasmid vector systems. They are the first, no, because they are the first type of vector systems developed and used in transformations in case of prokaryotic system. They are very much convenient, and people also try to extend them to the animal cells. Ease of subcloning is there in vitro manipulation and purification of recombinant proteins. For few of them, people have used these bacterial plasmid vector systems, and it's also have modular elements that can be included to drive the transgene expression. And example of some of the vectors which people have developed and used as a plasmid vectors are combination of virus and plasmid system. So these synthetic plasmid vectors are, you can say, semi-synthetic, or you can say derived vectors, are the PSV, which are based on the RSV they have used, which are based on SV40 and RSV virus. They have taken the transcriptional control sequence of SV40 and RAS sarcoma, and in order to develop these vector systems, and apart from the PBR1022 plasmid part, other kind of you can see these vector systems how they have used it, and they have added the marker gene, that is DHFR, and as well as NPTR, neomycin phosphotransferase gene, as a marker for selection. So based on that, and even GPT also was used. So based on that, people call them as SPV2, Neo, or GPT, or DHFR. And fourth property which was required is these should be replicative plasmids having an origin of replication which can operate in both animal as well as the bacterial systems. That was again a problem. So then people have gone for non-replicative plasmids, because in animal systems you need autonomic replicating sequences to be added, then only they can undergo the replication in case of animal systems. Whereas in case of bacterial multiplications, you required the origin of replication of for bacteria. So then they count for some non-replicative plasmids, because in animal, to make the plasmids to get replicated is a little bit difficult. So these non-replicative plasmids, they have one issue, that is you cannot able to get a stable transformations. They can actually provide only transient transformation, and that too with moderate recombinant proteins, and we can have the transformation for only 1 to 2 days. So to avoid these issues, people have gone for the other type of vector systems, that is they looked into the viral vectors. So these viral vectors are having a kind of positive effect in terms of transformation and in terms of the the DNA carrying capacity. So the advantage with the viral system is that due to their efficiency with which the virus can able to deliver the nucleic acids into the cells, and also a high level of replication as well as gene expression capacity, which is possible to by obtained by the viral systems, so viral vector systems. People actually thought of using different viral vector systems, means different viruses, and develop them into the vectors and use for transformations. There are four classes of viral vectors people actually developed in order to do the gene therapy, and these vector systems were retrovirus, adenovirus, herpes virus, and adeno associated viruses. So I'll be discussing only few of these different viral systems. Adenovirus and adeno associated viruses, they can enable the DNA fragments up to 8 kilobase pairs to be cloned into them, and longer than is possible with the whatever is possible with SV40 virus. But adenovirus handling is more difficult because of their genome is very big. So adeno-associated virus makes use of some of the proteins synthesized by the adenoviruses in order to do multiplication or for the replication cycle. So these adenoviruses genome insert into its host DNA in the absence of HSV, and particularly it inserts into the chromosome number 19. So adeno-associated virus are therefore looked as an having a major potential in the process of transformation. You can see this is a simple representation of adeno-associated virus genetic genome. So then people went into the other type of vector systems because they were also doing the transformation in case of insect cells. So that is, they get into the baculoviral vector systems. Baculovirus is one of rod-shaped virus having a larger double-stranded DNA genetic material, and it infects usually the insects and their cell lines. One of the group of the baculoviral vector is nuclear polyhedrosis virus, which produces a protein called nuclear occlusion bodies. The protein with the virion stable at even harsher conditions. The baculoviral vector are used mainly for high level of transient protein expression in case of insects and insect cell lines. People have used a protein in the baculoviral vector system called polyhedrin, so it's found to be produced in large quantity, and it involves in the formation of this nuclear occlusion stage. What people have done is they removed this non-essential of the means the proteins non-essential for infection. They have been removed, and particularly they use the promoter of the polyhedrin gene because it was very exceptionally strong promoter and producing large amount of mRNA. Almost 25% mRNA is going to get produced by this particular gene promoter. So by using that combination, people have produced two sets of baculoviral vector systems. One is called as they have used two baculoviral baculoviruses for developing into the vector system. That is Autographa californica multiple nuclear polyhedrosis virus and Bambix mori nuclear polyhedrosis virus. And in insects, post-translational modification is going to be a little tedious. There is particularly glycosylation, and even whatever the protein which people actually developed in the insect cell lines has an issue. Means when that particular proteins were used into the animal system, there was a problem of uh, developing immunity or immune response against to these particular proteins due to the issues about their structures and even the glycosylation. So this has been addressed later by using insect cell lines specifically for their ability to carry the mammalian type of post-translational modifications. Example, people have developed cell lines from Estigmin acrea, and they have used this promoter polyhedrin, and inactivation of polyhedrin was considered by insertion of foreign DNA in between this particular polyhedrin gene, and use of helper vectors also done because whenever the size of the vector becomes very high, since it was unable to carry all the genes required for infection as well as expression and insertion, they've used these particular genes onto the other vectors called helper vectors, and later after insertion means after transformation into the same host by through co-transformation, they developed a recombinant they made them to undergo recombination and develop into a larger vector. So it can lead to the size problem, and they solved it by deletion of non-essential regions. So this is one vector systems which are developed by deletion of non-essential regions, and develop this vector system. Then another type of vectors which people used is vaccinia vector or vaccinia virus was used as one of the most widely used vector system it is. So vaccinia virus is closely related to variola virus, which is responsible for smallpox and used as vaccination. Uh, the success of this particular virus led to the idea of constructing recombinant DNA, means recombinant vaccinia viruses. The pox viruses have a complex structure and large double-stranded linear DNA molecule of about 300 base approximately. And this is how the people actually developed their vaccinia vectors. They used truncated vaccinia viruses, and they made them to infect with the host having bacterial plasmid carrying the recombinant, the bacterial vector carrying recombinant DNA. They made it to infect to the same host. So once they both infected the same host, there was an homologous recombination happened between the vaccinia and the plasmid DNA, which results in the formation of a recombinant vaccinia virus carrying the TK negative gene, means inserted gene. So that made the viruses as a vaccinia virus, and then that recombinant virus was used for the transformation. Another way of constructing the infectious vaccinia virus by expressing the influenza HA gene was like this. So vaccinia virus, which is thymidine kinase gene inactive, inactivation done by insertion of the fragment carrying the early gene promoters, and then on the other hand they have taken the restricted influenza HA gene, and both were actually means by cutting the TK gene by using BAMH1, they have inserted the fragment of HA into that particular site and then ligated it, and by the process of artificial means by the process of transformation, so this particular recombinant vaccinia DNA was made to get transformed into the host cell, and it was infected with the vaccinia virus, which is TK positive wild type. And when this recombinant and wild type were both infected into the same cell, homologous recombination happens between the TK positive and TK negative recombinant vaccinia and as well as wild type vaccinia viruses, and results in the formation of a recombinant vaccinia virus carrying the TK negative and foreign gene into it. So like this, people have actually produced the vaccinia virus, and these viruses can be means it produces both actually wild type also, so which can be further separated by using by subjecting it for selectional pressure. Another type of viruses is retroviral vector. Retroviruses, they are based on the molynemurin leukemia virus. People actually develop them, these vectors, which were infecting both mouse and the human cells. The genetic material was studied and found there are viral genes like GAL, POL, and ANV. They are replaced with the transgene insert and expressed on the plasmid in the packaging cell lines. So just like the way vaccinia was developed, similarly they actually developed the retroviral vectors also. And because of the non-essential genes lack the packaging sequences, so they were not included in the virion particle. And to prevent recombination resulting in the replication competent retroviruses, so all the required genes of homology with the vector backbone was all removed, so that even if the cell is get infected with any kind of wild type retroviruses, there should not be any kind of recombination between uh, the vector and as well as the wild type. So this is a viral genetic material carrying the GAG, POL, and ENV gene, which are non-essential genes, and they have removed it. In place, they have put uh, the foreign DNA or the transgene, which you can say therapeutic gene, and then they are allowed to undergo package within a host cell called as packaging cell. This packaging cell will have the genes of GAG, POL, and ENV, which was transformed, means it was put into those particular cells into their genetic material, so that these particular proteins are going to get produced by these cells. So when the cell is going to get infected with this retroviral vector construct, lacking the GAG, POL, ENV for the packaging, so which will be sufficed, which will be actually complemented with the pre-existing genes in the host cell and produces the recombinant retrovirus, means packaged retrovirus, which can be used for transfection into a host cell. So the retroviruses are designed to enter the host cell and deposit the gene. Special vectors are constructed by deleting or altering the native sequences of retroviruses, and even lentiviral vectors are also produced in a similar fashion by preventing the generation of replication competent retroviruses. So transcription could be under the control of LTRs or enhancer promoters elements might be engineered with this particular transgene. The chimeric genome is then introduced into the packaging cells, which produces all the viral protein molecules such as GAG, POL, ENV, but these have separated from the LTRs and packaging sequences. So that makes the only chimeric gene to get assembled and generate into a viral vector, and it can be used for further transformation, and it can be with the culture medium in which these packaging cells are going to be grown is then applied to the target cells. The medium will have this particular protein, means recombinant vectors. This is how the retroviral vector constructs are done. You can see another retroviral vector constructions to make them to carry the target gene. How the packaging is done by the help of packaging cells. There are some limitations to the retroviral vectors. Critical limitations of retroviral vectors is their ability to infect the non-dividing cells. like muscles, brain cells, lung cells, they cannot infect it. And the cells from target tissue are removed, grown in vitro, and infected with the recombinant vector. The target cells are producing the foreign DNA protein are then transplanted back into the animal cells. Like that, people actually did it. So, in order to solve the problem, and the expression is reduced by inflammatory interferons acting on viral LTRs, and as retroviral DNA integrates and viral LTR promoters are inactivated. So there are some kind of some of the means limitations. Advantages wise, the capacity of insertion and creating a permanent expression or creating a permanent cell lines at stable transformants. Pseudotyped viruses are again advantages of it, and whereas disadvantage wise, it can infect only dividing cells, and insertional mutagenesis is again a problem. It can form tumor formation can takes place if we use it into live for gene therapy, but to develop cell lines it can be helpful. Another type of vector systems which actually people develop is lentiviral vectors that belong to the retrovirus family only. So they are more complicated than retroviruses because they are having additional 6 proteins required for virulence, and HIV is been disabled. They are using HIV also as a vector system by disabling the virulent genes and using it for in vivo gene delivery. So because it develops low cellular immune responses and has good possibility for in vivo gene delivery, so no potent antibodies are going to get produced against to this. So lentiviral vector systems is similar to the other retroviral vector systems and is comprised of transfer vector and that carries the transgene and a packaging cell line which provides the viral protein molecule. So viral vectors are usually successful to introduce a variety of transgenes into the host cells, into the embryonic cells and early embryo hosts, and all people have done it by using this lentiviruses. So the transgene is with an appropriate promoter sequence is inserted into the lentiviral vector. So the transfer vector is introduced into packaging cell, produces the viral protein molecules required for production of viral RNA, and produces the proper recombinant lentiviral which can be used for infection. So long terminal repeats and at the 5' 3' end of the vector are going to be required for production of lentiviral RNA, and packaging signal also is going to be required for packaging of RNA. And people have used this PPT polyurine tract sequence and would check post-transcriptional regulatory elements to enhance the transduction of host cells and increase the transgene expression in the animal cells. The regulatory elements within the 3' LTR is deleted to prevent the production of viral RNA from the promoter which are present within the LTRs following means once it is introduced to the host cell, so that only the transgene is going to get developed and it's not get affected by the deletion. So there's a comparison. Sorry, this table has not properly obtained. Later I'll give you all the PPTs which will help you in coming across with. So there are viral varieties, like people have gone for using adenovirus, retrovirus, herpes, adeno associated, and they have different capacity of transformation, 10 power 11, 7, and like that. Some of them, few of them are very good producing transformants and produce high transformation rate. In case of adeno, others are going to be having less. Okay, so these are all some viral vectors which people actually developed and used it for the transformation in case of animal systems. So I'll stop here. In the next set of video I'll be sending how different methods have been employed in transformations. Thank you.